The last category of macromolecules that we need to discuss relative to nutrition will be the proteins. They have numerous and extensive important functions in the body, and we will be going over those in another video, so stay tuned on that. Everybody knows that meat is an excellent source of protein. There are plenty of other foods from which you can get adequate amounts of protein, including dairy, including beans, as well as grains and vegetables. And we'll talk about complementary proteins in another section. You know what I'm going to say about all the videos that are posted? So watch them. Proteins are going to contain all of the same atoms as the other macromolecules. They will also contain nitrogen. It is the nitrogen in proteins that give them the name amino, which conveniently means nitrogen containing. And we know that amino acids are the building blocks, the monomers of proteins. All of the amino acids are going to have the same basic structure. There will be a central carbon, there will be hydrogens hanging off that, and there will be an amino group, NH2, as well as an acid group, chemically that COOH, attached to that central carbon. Because carbon atoms are required to form 4 bonds, you need to have a 4th bond that is attached to something else. It's that 4th bonding site that is going to make every amino acid unique from the others. That 4th attachment is going to be associated with a side chain, or what's known as an R group. So that side chain is called an R group. And that just shows you the central carbon. It shows you the amino group on the left. It shows you the COOH on the right. That's a carboxyl group. And then that side chain is shown by that letter R. Those R groups will be different for each amino acid. That means that proteins can be highly complex and will have multiple functions, many more so than either carbohydrates or lipids. A protein is pretty much always made up of varying types of amino acids, and each one is going to have some of those different side chains, or one of those different side chains. And there are going to be 20 different side chains, which means you're going to have at least 20 amino acids. The simplest monomer is going to be glycine. It has a single hydrogen atom, has its R group or its side chain. The other amino acids will have more complex side groups, which means they will be different in size, in shape, in charge, and in function. That shows you a guide to the 20 common amino acids. Just take a quick look at that. No, I'm not going to put this chart on an assessment and say, ooh, quick, identify or tell me which one is aspartic acid, or tell me which one is phenylalanine or tryptophan. This is for your reference only. Sorry. More than half of the amino acids are considered non-essential, which means that our liver, our amazing liver, can synthesize them. Protein in the food that you eat often supplies these, but it's not essential that you eat them. If you have nitrogen available to form that basic amino group or that base amino group, and you have fragments from either carbohydrates or fats, the hepatic cells can synthesize any non-essential amino acid. There are 9 super important amino acids that we can either not synthesize, or we cannot synthesize in large enough quantities in order to have sufficient amounts to meet your metabolic requirements. That means that those essential acids are essential, which in turn, you'll remember, means they have to be supplied by the food that you eat. Sometimes a non-essential amino acid can become essential because, here's an example. Generally, or often, we normally use phenylalanine, which is an essential amino acid, as a base. And that base is used to synthesize tyrosine, which is not essential. If you are not consuming enough phenylalanine, the scientists and researchers don't know why, but if you cannot make that conversion, then tyrosine actually becomes what's considered conditionally essential, which means you're going to have to get it through the food that you eat. Our cells are going to synthesize proteins by combining amino acids together, and the way that those can be combined together seems almost infinite. That means you will be able to make thousands and thousands of various types of proteins. Each amino acid is going to be joined to another amino acid by a peptide bond. And if you remember anything about biology and the functions of the cell and the cell organelles, you will remember that binding together or bonding together of amino acids happens at the ribosome, which are in the cytoplasm, through those condensation reactions through dehydration synthesis. So you remove a molecule of water in order to create a bond in between two amino acids. And then you remove another water molecule to combine one of those amino acids with yet another. So you end up with what's known as a polypeptide chain. A polypeptide chain. How does that happen? You take a whole bunch of amino acids, you have them undergo dehydration synthesis by removing a molecule of water, and you end up with a polypeptide chain. It is the number and sequence of amino acids in that polypeptide chain that forms what is known as the primary structure of a protein. I will repeat that. This is going to happen at the ribosome, which you'll remember is within the cytoplasm of cells. It might be actually attached to the rough endoplasmic reticulum. Holy cow, are you thinking back to your basic biology? At the ribosomes, amino acids will be strung together through dehydration synthesis. When you have sufficient amount of those amino acids bonded together through dehydration synthesis, you have formed peptide bonds in between each of those amino acids, and you have created a polypeptide chain. The primary structure of an amino acid is a specific number and a specific sequence of amino acids bonded together by peptide chains, by peptide bonds into a chain. If you think about a string of beads, multicolored beads, that's kind of sort of a great analogy for a polypeptide chain. So if you have two amino acids that are connected together through dehydration synthesis, you have a dipeptide. If you have three that have been connected or bonded together through dehydration synthesis, you have a tripeptide, and you end up with a polypeptide chain when you have multiple amino acids that are attached together through dehydration synthesis. Most of the proteins in the body are at least a few dozen, if not several hundred amino acids long. So that primary structure of a protein, those polypeptide chains, can be incredibly long. Make sure you watch that. So we've already talked about the polypeptide chains. They can twist into a whole variety of complex shapes, depending on the sequence of those amino acids. Those side R groups are going to be responsible for forming the shape, the structure, and providing the electrical properties, as well as the function of those proteins. Make sure you watch that. That's an excellent little video. So let's take a quick look at this image. You can see on the left, it shows you what the primary structure of a protein looks like. You have a whole bunch of amino acids that are strung together in what looks like a chain of pearls or a chain of beads. If you take that primary structure, we're looking at the diagram on the right now, you can see that polypeptide chain and see how it sort of turns into either a helix or it folds back on itself. When you have that helix or that folding back, which is known as a beta pleated sheet, you have what's known as the secondary structure of a protein. Silk is actually a secondary structure protein, so that's why silk is so unbelievably strong. If you take that secondary structure and you sort of fold it up all together in a mess on itself, as shown in the picture, sort of towards the bottom on the right, that is the tertiary structure of a protein. And then last but not least, if you have two or more of those tertiary structures and you attach them together, you bond them together, as shown in the lower left image, you have what's known as the quaternary structure of a protein. By the time you get to a quaternary structure protein, you are talking about an enormous compound. They are huge, relatively speaking. We're talking about something like one example is hemoglobin. Hemoglobin is big enough to carry 4 oxygen molecules. And if you remember anything about oxygen molecules that's O2 is the chemical formula, which means there are 2 oxygen atoms in an oxygen molecule. So hemoglobin is a quaternary structure protein, which is large enough to carry 8 oxygen atoms for oxygen molecules. The last thing I'm going to say in this section is this, and write this down so you remember it forever. When you change the structure of a protein, you change its function. When you change the structure of a protein, you change its function.